and then following we'll have Leslie and Chrissy. Ahead.
I had one question.
Radhika?
Do we have a practice problems for protein modification, or there would be none in the next week?
I don't think I have a set. I can look into my set of practice problems, but I don't think we have a set.
Okay, I was just curious and planning my work ahead, but thank you.
Yeah, I will double check, but I don't think so. Okay, Laura, you can share your screen.
Okay. Pam, can you see my slides?
Yes.
Okay.
And Laura, are you your first presenter?
Yes. Or no, sorry, Paul is. Paul is.
Okay, so presenter one is Paul. And okay.
Laura, now you said you were going to do the introduction part.
Yes, yeah, he's presenting on the first paper. That's what you mean by presenter one, right, Pam?
Okay.
Oh, it doesn't really matter. I just wanted to write the names for name one and name two based on who's going to start first.
Oh, then I start first. We'll just sorry, everyone. Just put whoever you want. I'll start first, though.
Okay.
All right, ready, Pam?
Yes.
Okay. All right, well, good afternoon, everyone. Today, Paul and I will be presenting on the use of protein modification methods to reduce the allergenicity of soy protein. So in 1999, the FDA approved a health claim connecting soy protein with heart health, which has increased the use of soy protein ingredients in a variety of foods. However, soy protein is one of the big 8 allergens, which cause 90% of food allergic reactions in the US. And those food allergic reactions can produce symptoms ranging from hives to anaphylaxis in severe cases. And currently the treatment for individuals with a food allergy is to avoid that food completely. So let's start with a quick overview of how the body responds to a food allergy. So after your body has been sensitized to an allergen and you're subsequently exposed again, the allergen binds to IgE on a mast cell, and the cell releases histamine and other chemicals that cause those allergic symptoms. Epitopes are the specific sites on the antigen, which in this presentation, when we're talking about antigens, we're talking about soy protein, that bind to the IgE antibody. And those epitopes can be on the exterior or the interior of a folded protein, and there's a variety of possible epitopes in a protein. So from class, very familiar since we were just tested on it. Not that long ago, we know soy protein is made up of globulins and albumins, and that the albumins are mainly enzymes and structural proteins. We know that globulins are made up of glycinin and beta-conglycinin, and that these seed storage proteins provide functionality to food systems. But unfortunately, they're also likely the proteins responsible for causing an allergic response. So as we're going through and talking about how we're trying to alter the proteins to reduce the allergic response, we're also talking about altering the proteins that provide the functionality to the food system. So the 2 papers we'll discuss today both try to reduce allergenicity of soy protein through protein modification. The first paper is going to use enzymatic hydrolysis, while the second paper is going to use enzymatic hydrolysis followed by Maillard-induced partial glycation. So as we talk about both these papers, we'll refer to the degree of hydrolysis, or DH, which is the percent of peptide bonds hydrolyzed by an enzyme.
All right, so modifying with proteases can alter functionality, such as improving solubility and other functional properties. It can increase allergenicity through exposure to isotopes, or it can decrease it by destroying epitopes. So far, producing a non-allergenic hydrolysis would require a completely hydrolyzed protein. It takes 90% of hydrolysis or more to see reduced allergenicity. At the same time, each individual's allergenicity is different. Glycation has been shown to reduce the quality and digestibility of soy, and it can block lysine, an essential amino acid responsible for cell growth and repair. Previous in vitro testing has not shown a lot of correlation with in vivo testing. It doesn't take into account cross-reactive carbohydrates, low-affinity compounds, and lab tests may only use one epitope. Investigation with human hosts would be a better representation of allogenicity. My paper is the enzymatic hydrolysis does not reduce the biological reactivity of soybean proteins for all allergic subjects. Next slide. The authors evaluated soy protein isolate treated with various enzymes and in vivo. After obtaining 9 human sera samples, a component of blood, they hydrolyzed SPI with, if you remember from class, the generically named alkylase, papain, bromelain, trypsin, and chymotrypsin under the ideal temperature and pH for each enzyme for 5 to 60 minutes. They ran electrophoresis in reducing and non-reducing conditions, incubated the gels with the diluted sera, followed by 2D electrophoresis. A mediator release assay was done by adding immortalized rat leukemia cells as a host for the gene responsible for IgE production. Resulting release of a compound known as a mediator signaled the immune response calculated as a percentage of total IgE produced. Identified some resulting fragments using mass spectroscopy as well, but for the sake of time, suffice to say they found pieces of glycinin and beta-glycinin. Next slide. These gels contain samples of hydrolyzed soy protein isolate with the lanes numbered according to the key shown here. Trypsin and chymotrypsin were run on the same gel. The controls were heated SPI, which was SPI incubated in each enzyme's ideal temperature, and unheated SPI, which was simply diluted in saline, followed by each enzyme at various time points of hydrolysis at 5, 15, 30, and 60 minutes. Controls revealed many bands between 10 and 100 kilodalton, and a common theme was new bands around 6 to 10 kilodaltons after hydrolysis. Oddly, there was no lane 7 at the alkylase gel, noted by the question marks here. This was not addressed in the paper. The authors noted the majority of hydrolysis also had occurred by 5 minutes with not much change after that. They'd added 20% solutions of SPI to each lane, but failed to state whether they accounted for the actual amount of protein added. And the lower molecular weight bands should be darker, which suggests that they can't really make a claim about hydrolysis efficiency if there are contaminants present. Next slide. They ran an IgE immunoblot on 3 different sera, but they focused most of their discussion on one sera. Bolder bands here mean greater binding of IgE. The increased bands at 20 kilodalton could have been new epitopes or smaller fragments of large bands already responsive to IgE. The authors acknowledged papain had a significant reduction of bands, but they didn't really explore it any further than that. Recall there were 7 total lanes at electrophoresis, back to last slide, representing the 3 controls and 4 time points. For this immunoblot, they only did 6 lanes. They removed the 15-minute time point with no explanation. Next slide. As with the other enzymes, trypsin showed many higher binding 20 kilodalton bands. All right, next slide. The 2D electrophoresis included SPI treated with alkylase, trypsin, and chymotrypsin only for 60 minutes. So they eliminated a couple enzymes and their lower time points. It's not clear why they chose to do that, but perhaps it was due to the high hydrolysis, even though 5 minutes they stated would be sufficient. Bands larger than 37 kilodalton had generally disappeared. Alkylase revealed faint bands between 12 and 25 kilodaltons, with many more obvious bands treated with trypsin. Numbered spots in these gels are spots that were found in both controls and hydrolyzed samples. The 2D results had what appeared to be smearing in the control and chymotrypsin samples. Nothing was said about that. Numbered spots, the spot 9 is a new spot that was not previously seen before. Mass spec revealed in general alpha prime, alpha, and beta subunits of beta-glycinin, but they really didn't discuss the identity of spot 9. So again, that's a new spot that we don't know what it is. For the mediator release assay, this was a result of IgE binding to basophils, which is the rat cells, resulting in the release of mediators. In this case, the mediator was beta-hexosaminidase. They included the enzymes alkylase, trypsin, and papain again. The heated SPI controls showed greater or less response than many of the hydrolyzed and unheated samples, even though aminobots showed comparable results. This could mean the presence of digestion-resistant proteins. The authors attributed the higher release to the 20 kilodalton binding seen earlier. A couple things to note are the inconsistent y-axis on each graph, which could be a result of Excel automatically assigning a value, but that could have easily been changed. And the unheated control, again, only have been dissolved in saline, showed a wide range of responses to each enzyme from 40 to 60 micrograms per mil, when it would be assumed that it wouldn't be so much variation. Next slide. Some limitations noted by the authors is that they only had 9 total donors, and only 1 had enough serum that could be used in the mediator release assay. And a few of the ones they did acquire were not at all allergic to soy. And the donor and that fresh blood is needed for mediator release, but again, the rat cell lines could be a replacement for that, but ideally you're using fresh blood for these types of assays. And it's difficult to correlate the IgE binding and basophil activity, mostly because they use that 20% solution basically as a guess of how much they should use. And conclusion, they concluded that few previous studies have evaluated biological response to specific epitopes. That you can find increased responses despite reduced binding. Allergic responses can still occur after hydrolysis. And foods should not be labeled hypoallergenic because of that. You still get the response. And what I found is interesting that alkylase and papain showed reduction and or disappearance of IgE binding, but there wasn't a lot of discussion about that. Next slide. For my review, I liked that they had several enzymes at several different time points. They chose enzymes that are currently used in industry for improvement of functionality. They compared in vitro and in vivo assays. They had a good comparison of pure soy protein isolate to serum responses. And they noted similar new epitopes. For areas of improvement, they had an overwhelming amount of data. They had several gels. All their gels were in 2 figures. So whenever they referenced back to figures 2 or 3, it was one of those gels, and I had to go back and figure out which one they were talking about. Beta-hexosaminidase was never mentioned until the very ends of their methods section. I had to go back and read again to make sure I understood that it was an example of a mediator. The mass spec, not really sure if it's worth including that at all, because it was all it did was find the obvious, pieces of glycine and beta-glycine. They would need to measure the protein content of hydrolysis and account for the actual protein in the STS pH of metabots. And of course, the inconsistent assay.
All right, our second paper is looking at the effects of limited hydrolysis combined with Maillard-induced glycation on the immunoreactivity of soy protein. And you will obviously recognize at least one of the names on this slide. And fun fact, the first author is actually my old boss from Cargill. So this paper used response surface methodology, or RSM, to determine which time, temperature, and enzyme to substrate ratios to test to produce different hydrolysates. So the general procedure for enzymatic hydrolysis is shown here using alkylase, which you'll remember was one of the enzymes researched in our first paper that showed promise in reducing allergenicity. Then they took soy protein isolate and soy protein hydrolysate produced using the optimized conditions and subjected those to partial glycation following the procedures on this slide. And as a control, they had SPI and the optimized soy protein hydrolysate that were incubated at different time points without dextran, and those were also characterized. So this STS page gel shows the protein profile run under reducing conditions of the different hydrolysates. So when we're looking at the subunits of beta-conglycinin shown here at the top of the gel, the alpha and alpha prime subunits disappeared, and the beta subunit became fainter with increasing hydrolysis. When we look at glycinin, the A1, A2, A3, and A4 acidic chains became fainter with increasing hydrolysis, but the basic subunit, which is denoted by F, resisted enzyme degradation. And this makes sense because we know that the enzyme used, alkylase, is specific to hydrolyzing beta-conglycinin subunits. So these immunoblots examine the reactivity of the different protein hydrolysates using the sera from 2 different participants. The different components of beta-conglycinin and glycinin demonstrated reactivity, although it did vary between the 2 different participants. But overall, as we increased the DH, immunoreactivity shifted more towards those lower molecular weight proteins, which the authors hypothesized with that there was an exposure of more epitopes from the interior of the protein, or that new epitopes were actually being formed. So this graph shows the results of the ELISA assay for 2 different participants. So low DH value resulted in a low % reduction in allergenicity. And it was actually SPH10, which had a DH value of 6.77 that showed the most pronounced reduction across all participants. And we see as the DH value increases above 6.77 with the SPH 11, 12, 13, 14, and 15, the % reduction in immunoreactivity either remained the same or decreased. The authors then looked at the effects of partial Maillard-induced glycation on the hydrolysates, and the smearing on this STS page gel that you can see is attributed to the fact that glycated subunits can be glycated with a different number of dextran molecules. So from this, we see evidence of glycation of the beta-conglycinin subunits. And then compared with glycinin, we see that the beta-conglycinin subunits more readily participated in Maillard-induced glycation using dextran. Looking at the Western blots, they showed that the partially glycated hydrolysates showed less immunoreactivity than the partially glycated protein isolates. So immunoreactivity of beta-conglycinin was not necessarily seen in all of the blots with the hydrolysates. However, when looking at glycinin, there wasn't much of a difference that was noted in the immunoreactivity of glycinin upon glycation. So the authors noted here that it would be worthwhile to try using an enzyme to hydrolyze glycinin, such as using pepsin, in a future study to help with reducing the immunoreactivity like we saw with beta-conglycinin. So based on ELISA data, we see that partial glycation resulted in an additional % reduction in immunoreactivity for participant 5, but it resulted in an increase in immunoreactivity for participant 3. So these opposing results highlight one of the major challenges of this type of work, which the authors pointed out in the conclusions here. It's very challenging to evaluate reductions in protein allergenicity when human sera response varies from participant to participant to participant. So it's very challenging to look at that. Maillard-induced glycation could either help or hurt your goals at reducing immunoreactivity after hydrolysis. And they did note that RSM was a valuable addition to this study and would recommend doing it for applying that to Maillard-induced glycation conditions as well. So we felt that the strengths of this paper were that the reasons for research decisions were very well explained. It was written in such a way where they said what they did and then why, which made it a lot easier to follow along. Figures and data were well labeled. Protein content was accounted for in the different assays. Controls were used appropriately throughout, and then the use of RSM to optimize enzymatic hydrolysis conditions. Some of our suggestions, we would have liked to have seen the use of another enzyme to hydrolyze glycinin to see what that could have affected the results, which the authors mentioned as well. I found it a little bit challenging reading the SPH codes and going back and figuring out what the hydrolysis conditions were. And then according as the first paper noted, it would be beneficial to confirm those in vitro findings with in vivo tests such as a skin prick test or a biological assay, such as a basophil activation, just to confirm findings in human subjects. And then, since we're looking at trying to make something that's still functional, it'd be helpful to characterize the functionality of these resulting partially glycated hydrolysates. So looking at these two papers, our overall takeaways was that this work is really necessary. There's a prolific use of soy protein in the industry, and unfortunately, it's one of the big 8 allergens. However, antibody testing alone might not be sufficient to confirm the loss of allergenicity when we're doing these sort of tests. And after working on this, Paul and I kind of got to the end and realized, you know, is this even possible? Can we keep a protein and have it be functional and have it be safe for someone who has a soy protein allergy? So it's very interesting, very cool work. It's also very challenging because of how much humans vary. And with that, we've got references, and then we'll take any questions.
Very good. Thank you both. I think you kept your timeline. I think.
18 and a half minutes. I was
how many minutes?
19 and a half.
Yeah, I thought you were right on par. Okay, good. Yeah, questions.
I have a question with one of your conclusions. Did you, in your review of literature, did you come across any other modification techniques besides like glycation and hydrolysis that have been tested to reduce allergenicity?
So with looking at the intros for both of these papers, what they referenced and talked about, it was mainly enzymatic hydrolysis that was addressed and talked about. Since when you're doing that, you're kind of breaking down and trying to break down the epitopes.
I had a question on the first paper. So I was wondering why it was important that they collected the sera, and it said it was from individuals that were allergic to both peanut and soy.
Yeah, that's because the peanut has an IgE response as well. So it was basically to, I think just to account for that.
Okay.
I think it's cross-reactivity between peanut and soy. Isn't that right, Paul?
If you say so.
Yeah, because peanuts and soy, they're both legumes and oil seeds as well.
Oh, yeah, here we go. Yeah, that would do it.
Yeah, you found it, Paul?
Yeah.
Yeah.
Both legumes.
Yeah, so yeah, some patients that are allergic to peanuts won't be able to also consume soy and vice versa. Okay, I think there are a few questions in the chat. The first one came from Radhika to Laura. Laura, you want to read that?
Yeah, so Radhika's question, why is beta-conglycinin more reactive to Maillard reaction with dextran? I think with this, because the beta-conglycinin underwent hydrolysis, wouldn't there be more exposure of lysine groups for participation in the Maillard reaction? So then with the glycinin not really being hydrolyzed by the enzyme at all, it's got and I think I remember reading that one of the subunits also just has less lysine in general for glycinin. So.
Yeah, that's good, Laura. I mean, since we did the study, I know that we started off with very non-denatured protein, and with enzymatic hydrolysis, if the protein is hydrolyzed, it's also denaturing the protein and causing unfolding. So that is exactly right, Laura, exposing more of the lysine groups. Good.
I think the next one is Paul's question from Grace.
Yeah, so they did discuss
Can you read the question, Paul, please?
Did they report the degree to which the participants were allergic and how it may affect the results? So they recorded the symptoms that the people reported, such as anaphylaxis, throat swelling, and vomiting were some examples. And they also had rated the amount of IgE in their systems specific to soybean and specific to peanut. Then they did not include things like age or gender or nationality, if that would have an effect. They really didn't go a whole lot into the samples themselves. They noted that some weren't allergic, and they kind of had to cut down from 9 down to 3 down to 1 that were actually worth reporting on.
All right, I think the next one is for me from Kinsey. Did you choose your paper because you knew the first author? Yes, and with my background being nutrition, I found it really fascinating, the whole concept that we can make something less allergenic. And before reading this, I had no idea like how that would happen or how that would carry out. So I thought it was really cool to read more about like epitopes and then kind of bringing the biological side in with what we've been learning in class, but also highly because I knew the first author, yes.
And a fun fact, the first author has a nutrition background like you, Laura, bachelor in nutrition, and then came to us for food science for master's. So, Paul, the next one is for you.
Any need to study the kinematics of the enzymes? Perhaps. I'm sure someone will think of doing it eventually. And how they determine the threshold of allergic responses and how the concentration affects this. Is there a standard way to do this that most studies use? Well, I wouldn't know a standard way to do it. For the, I have to remember how they determine the threshold. Without taking some time to actually go through this again, they basically noted that there was very, very little response in the binding in some of the serum samples.
And I know they measured with like the sera samples in both of them, like the IgE, presence of IgE in them to confirm allergenicity. So they did have standard, like, I can't remember the exact.
Well, they had a positive control of each sample. This next one's for me
I've
too.
always wondered, I've only ever heard of like a threshold for gluten allergies, at least in testing food products. So I've always just wondered what they determined the actual level is for proteins.
Well, gluten is protein, right, Devin? So is that?
Well, I just remember like for actual like production, you can test, like if you want to say something's gluten-free based off the allergy, you can actually test for a specific concentration. But I've never heard of that for other types of allergies.
There is actually numbers for all of the big 8 allergens, but I don't have those numbers in my mind. But yes, in order to determine if it is free or there is a certain threshold for all of the big 8, and gluten is one of the big 8. So. Okay, there is a last question here from Holly.
Yeah, so about labeling food hypoallergenic, it's just that they made, in their last sentence, they said these results should provoke caution in those claiming to have produced hypoallergenic soybean food ingredients and stimulate more biologically relevant testing. So they aren't recommending to label anything hypoallergenic, and it's, to date, there really hasn't been a method to eliminate all the allergenicity. So they can't label it hypoallergenic.
Well, if we go back to your introduction information, you did mention, and we talked about it together, is that you need at least 90% or so of the peptide bonds to be broken. And then it can be hypoallergenic, such as, for example,
right?
Like in infant formulas, if you have whey protein in there and it's hydrolyzed to amino acids and dipeptides and tripeptides, they claim it to be hypoallergenic, and it's in fact hypoallergenic. So that's why your conclusions at the end, it's really hard to be able to have a functional protein that is hypoallergenic, because in order to get it to be hypoallergenic, you need to break down all of the potential epitopes, and it's very, very challenging work. So yeah, Another one came in. We'll take that one, last one here.
Just keep coming.
Got two.
Oh, we got two. We'll take Muath's one.
We'll take Moss.
Yeah.
Okay, I'll take it, Paul, since you did the last one. Did they mention anything about sensory analysis and if it might be affected? They didn't mention anything about sensory analysis, but like keeping the degree of hydrolysis under 8% is going to be helpful in limiting any production of like bitter peptides. But no, none of these had a sensory component to it.
Yeah, that's a good question. It's definitely another, other than functionality, looking at the sensory component for acceptability. So that's a good one. I just want to make a quick comment before moving on to the next presenter. I know that you put at the beginning in one of your introduction that glycation is contributes to the lower digestibility and blockage of lysine. So basically poor quality. This is debatable depending on the glycation, extent of glycation. So if the glycation is controlled or not controlled. So excessive glycation, yes, can definitely lead to reduced quality, but partial glycation should not. So basically when we block lysine, we make sure to not excessively block all lysine groups. And previous research in our lab, if partial glycation was done under controlled conditions, we actually saw 9% increase in digestibility. That means it caused a little bit of unfolding that helped the protein to be better digested. So there's a lot of controversial results out there, but it all depends on the degree of hydrolysis, the source of the protein, the source of the sugars. So I just want to kind of make that point, as we are going to discuss later glycation today, just to kind of defend the glycation modification. It's not really bad if we do it right. Okay, so that's just a general comment to keep in mind. I had one other quick question for Paul, actually. I don't know if I missed it. So papain showed a lot of reduction in immunoreactivity based on the IgE blood, immunoblot. But then when they did 2D gels, they didn't do papain hydrolysis. Is that correct? You're muted, Paul.
Jeez. Yeah, they mentioned papain being high reduction, just a couple of sentences, but then they just moved on to the next thing.
So they didn't explain why the 2D gels were focused on other enzymes.
Right. Yeah, it was weird. I would have liked to learn more about papain because papain seems to be doing the job. Let's figure out what to do with that.
Yeah, yeah. Okay, it was just an observation, and I was wondering why they didn't do that. Okay, we're moving on to our next presenter. So this took half an hour, which is right on time, and we'll have, yeah, it's all yours. So who's starting first?
I'll be starting.
Technically, yeah, Leslie.
Okay, go ahead.
All right. Hi, everyone. So today, Chrissy and I will be presenting on protein modification using cold plasma. So for our first protein that we're going to look at today is king prawn. It's also known as Pacific white shrimp. And overall, the proteins, which are myofibrillar proteins, have high biological value. And this protein accounts for 65 to 75% overall of the total protein in the prawns. So any structural or physicochemical changes can greatly affect the gastronomic properties.
Did we lose this?
What?
Kind of paused a little bit, right?
Is it back?
Yeah.
Okay. And the main categories of molecular weights that are in the myofibrillar proteins range from 20 to 100 kilodaltons. So proteins are very influential in determining the quality of the prawns. So their high susceptibility to deterioration during transport and storage can lead to faster protein decomposition and can have a large negative impact.
I think she froze.
Yeah, I think she froze.
There we go. She's still there. Cool. Okay, so the next protein that we're looking at is flaxseed protein. It's traditionally an oilseed, but it does have a protein content which depends on the variety. Fortunately, the authors of our paper did not give us a variety to reference, so we're assuming that it's 18 to 22% of our seed weight. There are two fractions of that protein within our flax seeds. It's a salt-soluble high molecular weight and a water-soluble low molecular weight profile. It has a really nice amino acid profile that's full of non-polar amino acids contributing to a high emulsifying and foaming capacities or capabilities. However, it has limited industrial use due to a large amount of mucilage polysaccharides that are in the hull and an inefficient extraction method, which leads to the presence and subsequent interference of those polysaccharides in the functionality of the proteins. So the topic of our presentation is about modification of these proteins. And so we're looking at cold plasma, specifically atmospheric pressure plasma jet. I've included a really dramatic and non-representative image. That's the one on that right there because it looks cool. However, the one on the left is a much more representative picture of what we're using for our samples here. So essentially in this cold plasma, we've got electrons being rapid fired through some gas. In our case, it's air, causing a bunch of high energy collisions that essentially rip your valence electrons off of the gas's atoms, which is going to give you a soup of, which we're calling plasma, of free electrons, free ions, and some neutrals or heavier matter. So this mixture is really cool for two good reasons. One, you've got your electrons, which have a pretty high temperature, but their relative density to the rest of that material, which is much colder, means that the temperature of the entire mixture is much lower. So that gives you a non-thermal treatment, which allows you to modify these proteins without damaging these proteins. So that's one, and the other is just it's a non-equilibrium mixture because of that different ratio of your electrons and your ions. So this plasma is suggested to cause conformational changes, induce some formation of disulfide bonds, etc., through oxidation specifically. So that's what we're talking about today.
So these are the papers that we're going to be talking about. The Prawn one, looking at the effect of APPJ on the properties of the proteins, came from the Journal of Food Chemistry, and Chrissy's paper on flaxseed came from the Journal of Food Science, looking at the physical, chemical, functional, and antioxidant properties. So here are two atmospheric plasma setups. They're very similar. The only main differences that we had were that the flaxseed paper used twice the flow rate of the air going into the plasma electrode, as well as the flaxseed paper also used a slightly lower frequency for the power supply. But both of them did in the end use the same distance away from the jet and the protein solution here. They both use 30 millimeters. We have this red flagged, as in the figure, we assume it was a typo that they put 35 millimeters because when they actually talked about their methods, it was 30 millimeters as described.
Sorry, so we'll talk about these a little bit more individually when we're talking about our papers, but just so that you have this set up here in front of you, you can see that the protein concentrations of our different treatments were very different. Leslie's used a 2% concentration and mine was a 0.5% weight by volume. Since Leslie's paper, or the shrimp paper, had much higher protein concentration, they also do a much longer treatment time, going from 0 to 10 minutes in increments of 2 minutes, while mine was from 0 to 4 minutes, mostly in 5 to 10, 15, 30 seconds. So we also are treating different amounts of protein by volume. The shrimp paper was 20 mils, while the flaxseed was 150.
So now we're going to go into our individual papers, and all modification. So the objective of this paper overall was to determine the influence of APPJ treatment on the structure and physicochemical properties of the myofibrillar proteins that were extracted from the king prawn itself. In the practical application of this research is looking at modifying the proteins in prawns to help preserve the food quality over storage and shipping of the fresh seafood to help prevent quality degradation. So for the sample preparation in my paper, they extracted proteins from fresh prawns. They used a very extensive series of different washings. From ground prawns, they washed and centrifuged in various different buffers. They used Tris HCl buffer initially, then they moved on to a potassium buffer. Then they did some more solubilization before they washed the pellet with potassium chloride, and then they did salt washes and finally dissolved the pellet in a different concentration of salt solution and then centrifuged with the supernatant being used for the treatments and subsequent analysis without any further modification, i.e. they did not dry down the protein. So overall, that purified protein supernatant was also adjusted to a protein content, like Chrissy mentioned, of 2%. And then underwent treatment for as a protein solution in airtight opaque bottles at refrigerated temperatures until they ran the assays. So for experimental design, anything that has been grayed out is something I'm not going to talk about due to time constraints. But overall, they looked at pH, protein solubility and turbidity using spectroscopy, foaming capacity through whipping with a homogenizer in a graduated cylinder, and particle size measured with a zeta sizer. They also looked
Leslie froze again. Leslie, you froze
Is
again.
it back yet?
Yep.
Yeah.
Okay. And then for statistics, they looked at ANOVA and Duncan's multiple range test, which, if you're not familiar with that, is similar to Tukey's pairwise comparisons of means. So for the first results that I'm going to look at, we're going to look at the pH, which in the overall study only ranged from 6 to 6.8. They found that pH was inversely proportional to the treatment time and experienced significant difference. But it did not reach its PI of pH of 4. So in previous research, they found that pH decreases more when plasma is applied to liquid matrices. So these results were something that they expected to see, as well as the reason of APPJ using air creates a lot of reactive nitrogen and oxygen species, which also includes some acidifying compounds such as nitric or nitrous oxide as acids, as well as hydronium cations. Next, we will look at protein solubility, and this is the solubility graph that they got. You can also see that the solubility of the protein significantly decreased as the exposure to plasma increased, which also was an expected result, as ozone is a strong oxidizing agent that is also produced by APPJ and can denature the protein, which leads to the loss of secondary and tertiary structures. And as a result, it can expose hydrophobic groups, which we learned in class favors protein-protein interactions rather than protein-water interactions to decrease the solubility. Looking at turbidity, it was also seen that protein turbidity increased significantly over the study, which again was attributed to polymerization of the unfolded monomeric prawn proteins, and it follows the solubility trend as well. When they looked at protein foaming, they saw no significant differences over treatment time with the foaming capacity. And this was something that was unexpected, as previous studies did note that there's usually a more significant increase in foaming expected. However, this was discussed as the ozone was a very strong oxidant and led to the proteins being excessively oxidized and potentially leading to them creating a very unstable foam. But again, this could have been confirmed if a foam stability test was run. can see here that there's a significant decrease in or increase in particle size. And this is again due to the plasma treatment leading to polymerization and aggregation of proteins. And this was something that they said was related to the oxidation of the proteins, again, by the reactive species that were in the plasma. Surface hydrophobicity also significantly increased with treatment time, which again corroborates that proteins were unfolded to expose more hydrophobic groups, especially phenylalanine and tryptophan residues were something that the paper mentioned as a possible reason as well with the oxidation. And finally, we're going to look at the secondary structure changes by circular dichroism. So this reaction, or this chart, is not very useful at all to giving any results. It's hard to draw concrete conclusions because they just gave us the spectra that they got. They didn't actually integrate the results. And usually you would want to integrate your results so you could determine the percentage of alpha helices in different beta structures and how those specifically changed over the treatment times, instead of just saying, oh, here's the untreated protein, this black line, and then, oh, looks like all secondary structure just disappeared, which would be untrue. It would change to other secondary structures. So finally, the critiques that I had were that research comparisons were made a lot to dielectric barrier discharge plasma, which is very different from APPJ, so I don't think it's a fair comparison. Additionally, I think more functional assays should have been conducted, especially since they're talking information. They should have also done foam stability, as well as potentially adding gelation for the textural implications that this might have in the quality of the prawn. And also the circular dichroism results, as I mentioned, should really have been integrated. So some recommendations that I made were potentially to have a more brief description of the protein extraction method or even modify the method. I saw some other papers had a very stripped down method for this, as well as just mentioning and coming back to the purpose of them doing this research. They never really came back to how this would apply to their purpose of increasing the shelf life or quality of the protein. So I think that they also should have done texture analysis and maybe even looked at whole prawns versus ground prawns and maybe treated whole prawns instead of ground. Additionally, one last thing that just bothered me is that their consistency in the bar graph shading was completely, it was so hard to look at, it hurt your eyes, and it had no consistency or rhyme or reason at all to it.
Okay, so moving on to our second paper, it's very similar. We're looking at physicochemical, functional, and antioxidant properties of flaxseed proteins. Which is the objective, with the practical application for these flax seeds would be to tailor these for use as nano delivery vehicles, which is on this slide. Will pop up eventually. There you go.
Sorry, my computer is slow.
No, it's okay. Keep going. Let's talk about pH.
Come on.
Oh, just kidding. We're going to talk about table preparation first. Okay, so with these flax seeds, they first subjected them to germination for 3 days. I've red flagged it because it seems weird. They cited it for an increase in synthesis and accumulation of phenolic compounds, but the paper is on proteins, and so we would expect that it would negatively affect protein concentration. Pam agreed, so I kept my red flag. But they took them, they germinated them, and they moved into our isoelectric extraction, essentially defatted, adjusted pH to 9 sodium hydroxide, centrifuge, took it to the isoelectric point, precipitated it, freeze dried it, and did a crude analysis. They then took that powder, they redispersed it at 0.5% weight by volume in deionized water to pH 9, stirred it for 4 hours at room temperature, and then stored it overnight at 4 degrees Celsius. They then treated with the atmospheric cold plasma. Again, it was 150 mils for 0, 5, 15, 30, 60, 90, 120, and 240 seconds using compressed air. Now we're going to talk about pH, I hope. Nope, still not there. Okay. Similar to Leslie, we're looking at pH, turbidity, protein solubility, and some secondary structure. I'm going to talk about the same parameters that Leslie did, and also our functional properties, foaming activity and stability, emulsifying activity and stability, as well as statistics. finally here, team, we're going to talk about some pH. We're seeing, similar to Leslie, a decrease in our pH, which is expected. It's largely due to that generation of nitrous and nitric acids and the hydronium ions. And it's been validated by other research. I do want to call attention here to your 90-second mark because the pH of flaxseed is 4.2. And so with 90 seconds, our pH has descended to our PI of that protein. And we're going to see a lot of effects of that on our coming slides. Okay, so looking at turbidity and solubility, our graph on the left is turbidity, graph on the right is solubility. We see the decline of the pH in the previous slide, and so with that, we're likely going to see a collapse of our spatial conformation of flaxseed proteins and exposure of the hydrophobic groups, which are usually retained on the inside of the structure, as we know. So when that happens, our proteins are going to aggregate, and we're going to see a decrease in solubility. That is reflected here, as we see. That's also going to be reflected in our increase in turbidity. Again, I'm pointing out that 90-second mark because that's where we start to see really significant changes, which tells us here that our authors did not probably correct the pH back to the control level before doing these sorts of activity activity analyses. So that's got a red flag on this because that is not the proper way of doing these analyses. But we're going to keep talking anyway. Now we're going to look at circular dichroism. I had to look this up, so I'm going to let y'all know that it uses circularly polarized light to elucidate your secondary structure. If we cared about this graph, which we don't for similar reasons that Leslie mentioned, we would be looking at this 200 to 260 nanometer wavelength where we're seeing that absorption and emission. We would see that there's high fluorescent activity at our lower treatment times, and it gets quenched with the longer treatment times, however, which would be attributed to a conversion from your alpha helices to your beta sheet structures. However, they don't actually do any sorts of integration or elucidation of whether or not that's actually happening. They just give us this graph. And so we don't trust it, hence that red flag. Moving on to look at our foaming and emulsification capacities. We're seeing a light increase here at our short times for both of these. My paper also looked at capacity as well as stability, which good for them, thumbs up. You're seeing a light increase with your short times due to that increase in surface hydrophobicity, which is going to give you a more flexible protein structure, which is going to be able to be absorbed at your air-water interface. And then that decrease is because of the formation of your insoluble protein aggregates due to your excessive unfolding. So this, like when they were discussing this, this was also the only time in the paper that they mentioned those nano delivery vehicles again. And it was tangential at best. The foaming capacity and stability reflect the capacity of the protein to form and maintain thin films at your gas liquid interfaces. So with that in mind, we're going to see that the shorter treatment times are more relevant for those purposes, and same for emulsification. So the authors here are going to conclude and tell us that APPJ treatment is going to give you your decrease in pH and solubility, and your concomitant increase in your hydrophobicity and oxidation. With your shorter treatment times, you're going to get better foaming and emulsification properties, and thus is going to enhance your functional potential of your flaxseed protein for use of nano nutrient delivery vehicles. But we're going to move into my critiques, which are many. I had some issues with all of their methods. They didn't do SDS page ever, so we don't really know what's going on with polymerization or anything like that. Their CD integration is non-existent. There's that we didn't look at our pH adjustment. Also, this isn't necessarily a critique, it's just a thought that I had from my previous work in my other lab at the University of Michigan, is this idea of whether a 0 time point can count as a control, if those are interchangeable or the same. So not a critique, just kind of something to think about. They don't give any background on nano delivery vehicles, which means that we don't really know if they achieved the results that they were looking for. I don't know which of these changes were favorable or not based on the paper itself. Also, similar to Leslie, they were comparing their APPJ treatment with non-standardized methods. As by that, I mean like this difference in protein concentration, 0.5% to 2%. That feels like it would elucidate or I don't know, elicit quite a large change in your results. So it seems fishy. So recommendations would be to fix all of those things. We're going to adjust the pH, run some SDS page, maybe look at gelling capacity. That would be great, and do some secondary structural change analyses.
So overall, both of our papers did find that cold plasma does have an effect on protein structure. However, we weren't able to really make conclusions if this was something that would accomplish the goals that were stated in the introduction of each paper for the purpose of the research. So that's something that we'd want to investigate further. There are references. Are there any questions?
Okay, I have a question on the percent protein. So I think actually you're comparing 0.2 and 0.5% protein if it's 2 milligrams per mil.
That was my comment. Yay, Allie.
Yeah, but just regardless of that, even that seems like a really, really tiny concentration. Like I know in our lab, at least, like Sam did higher concentrations. Do you think that that has implications on the results? And I don't know, it seems like this wasn't so applications focused.
Yeah, I mean, also a lot of the papers that they were looking at like 1% protein, and they did actually have 2% protein. So all of the things that we're referencing and citing have a lot of different protein concentrations. I guess they do see sort of similar trends in the thing. So perhaps it doesn't have that much of an impact. Leslie, you're welcome to chime in here as well. But I don't know, it seems common sense that it would have an effect to me.
I agree with what Chrissy said. I'm not sure why there is such a big difference.
So, but just to clarify, you guys thought that they did at 2% protein? Did you do the math to conclude that it was done at 2%, or they reported it at 2%, but it's 2 milligrams per ml, really?
I think at one point I had the math calculated. I'm not sure. I think that got lost in our slide modifications, but I did have the actual grams per protein solution. Like in this slide, I had it calculated out at one point. There was still a difference between two of them. Yeah, I'm not sure the 2% looking back is correct.
Yeah, if they stated that it's 2 milligrams per ml, that's 200 milligrams per 100 ml. So that's 0.2%.
Yeah.
Yeah. Okay, so there are several questions that came in the chat. We'll go with the first one. It is for Chrissy.
Oh, yeah, it's a nanodelivery vehicle. Wonderful question. I had to look that up too. Essentially, you can conjugate a drug to a molecule, a lipid, a protein, whatever you're looking for, to enhance the delivery of that drug into the body, because they're more bioavailable. Also, they can protect your drug from degradation by your body because it's attached to something that your body does want. So flax is really great for that because it has lots of good things that you want. It's got high fiber, it's got good antioxidant compounds, it's got like anti-diabetic and anti-cancer sorts of functionality. So your body is going to welcome that molecule rather than rejecting it.
Okay, there is a question for Leslie.
Yeah. So Laura asked if they said in my paper how long it was between the protein extraction until analysis, or if they did not chose, or if they mentioned why they chose not to dry and freeze the proteins. So for the first part of the question, they did not say how long it was between the protein extraction until analysis. That was something I had questions about, because no matter how well you store it, there's still going to be some degradation since you broke down the proteins. They did make the point to have an entire section of the paper devoted to how they sourced the fresh prawns from a local supermarket seafood section. It was a Lotus supermarket in China. And then they said that they got it.
Oh no, we lost Leslie.
She's back. You're back, Leslie.
Okay. They made an effort to really get the prawns back to the lab as fast as possible. So I would think they tried to do the analysis as soon as possible after preparing the proteins, but they didn't actually mention. And they also did not mention why they chose not to dry and freeze the proteins as well.
Yeah, that's potentially a problem because they're running the assays and we don't know at which concentration they're running the assay. They didn't specify that. So this is a big flaw, I thought. Another comment about the big flaw, and I don't know if you guys mentioned it or I missed it, is when the pH drops and it goes down to the isoelectric point. Of course, you're going to see changes in structure and functionality, and the changes might not be attributed to your plasma treatment, but more so to your pH changes. So in work we've done in the lab, we notice a decrease in the pH. So we kind of adjust the pH prior to the treatment and adjust the pH after the treatment to neutral so that we can really extrapolate the impact of pH and be able to assess the impact of cold plasma. So I guess a lot of these things we're seeing in the paper, it is to kind of open your eyes towards how you design your experiments and how to critically be able to analyze your data without having anything masking the impact of a certain treatment, such as the pH masking the impact of cold plasma. All right, so we have a few other questions here. So there's Grace to Chrissy.
So what would I recommend as a control for the authors if they were to run it again? It's a really great question. Can be definitely hard to answer and depends on what you're looking for. So like Leslie's is maybe a little bit easier to answer. She mentioned running ground versus the whole shrimp, which would be an interesting comparison just because since hers is looking at shelf life, right? So that's a good one for that. For flaxseeds, it's a little bit harder because I'm not really well versed in nano delivery vehicles, but like I guess it depends on the treatment and what exactly you're trying to gain. Here, I do think that the 0 is probably a good control for that situation. But yeah, I mean, it sort of just depends on your methods and what exactly you're trying to improve, if that makes sense. I know that that's not exactly the best answer, but I hope that it sort of answers that.
Okay, we'll move to Mua's question to Leslie.
So in turbidity, the results showed an increase. And what does that mean? Is it good or bad? So going back to the turbidity, the increase in turbidity just means that the proteins are polymerizing, and as solubility decreases, they're increasing the turbidity of the solution just because they're coming out of solution and precipitating.
Leslie, we lost you again. All right, so while we get Leslie back, let's see if the last question is also for Leslie. Leslie, you're back. Are you back?
What's happening to her connection?
I have no idea what's going on with my internet.
Okay, so Leslie, maybe because we want to move along, we'll give points to Holly and Redika for asking questions, but can you type the answers in the chat and everybody can read that? But I want to give folks 5 minutes break and come back for final portion of the class. All right, we'll reconvene around 4:07. Okay, let's come back. All right, do we have everybody back? I know it's not a long break, but don't want to keep you long either. So we still have Nigel, Radhika, Wath, Kenzie. All right, I'm just gonna get started. All right, so let me share my screen. Okay, so this is where we left off last time. Can you guys hear me well? Okay, let me know if I turn robot because sometimes I do after a while. Okay, let me share my screen here. I shared my screen, I mean project. Okay, so I didn't prepare recap because I know we don't have a lot of time. So I want just to continue where we left off. And I started talking about hydrolysis as a protein modification last time, controlling the hydrolysis, how does different conditions impact the hydrolysis, whether it's degree of hydrolysis with the percentage of peptides broken, or hydrolyzed, controlling it by the source, whether the protein is denatured, non-denatured, the specificity of the enzyme used, and the optimal conditions for the reaction to occur, pH, temperature, and time combination, as well as protein to enzyme ratio. So enzyme modification kind of if it's controlled, it can cause targeted enhancement in functionality. And theoretically, when a protein is hydrolyzed and you get peptides released, you get more ionizable groups released. So you have more of the free carboxyl and free amine groups. So you have more charged groups that will interact with the water. So theoretically, you have enhanced solubility. So it's not just ionizable groups, but also shorter peptides tend to interact more with water than larger polymers. So theoretically, you have enhanced solubility. Also, with enhanced ionizable groups released upon the hydrolysis of the or breakdown of the peptides, you have thermal stability because you have more interaction with water and less interaction, protein-protein interaction. And more interaction with water, potentially you have more better water holding capacity in terms of if you form a gel, due to potentially enhanced balance between repulsive and attractive forces. So again, enhanced surface activity. But all of this is theoretical, right? So it's not always going to happen. It all depends on the nature of the peptides released. So this is theoretical, but sometimes we release peptides that contain more hydrophobic residues in terms of a percentage of hydrophobic residues than polar residues. So that could shift the enhancement in solubility. For example, it might be decreased solubility. So this is all theoretical. It depends on the specificity of the enzyme and the peptides, the nature of the peptides and the composition of the peptides released. Okay, so because it's a complex way of modifying the protein, so because there's so many different outcomes that we can get, so in order to kind of get to a targeted enhancement, we need to figure out what's the best conditions of hydrolysis. So you have different sources of proteins, you have different level of denaturation of the protein, you have different impact of environmental conditions. So how do we get to what's the best pH, what's the best temperature, what's the best ratio of protein to enzyme, what's the best time of the hydrolysis, and other conditions such as the nature of the protein and the state of the denaturation of the protein prior to the hydrolysis. So we have too many factors, too many independent factors that impact that would impact the results of the enzymatic reaction. So in order to get targeted improvement, for example, improved solubility in this example here, we use statistical designs such as response surface methodology. In response surface methodology, so instead of doing full factorial design, so for example, if you want to have 4 independent variables, let's say enzyme to substrate ratio, pH, time, and temperature. So 4 independent factors that might impact the outcome of the hydrolysis. So if you want to have full factorial design with 3 levels, like 3 different times, 3 different pHs, 3 different temperatures, or enzyme to substrate ratio. So if we want to have full factorial design, you end up having 81 experimental units. Imagine you sitting in the lab and running hydrolysis 81 times. That's next to impossible. If you're a graduate student, you will never graduate. So, and also if you're working in an industry and you want to figure out the optimum condition, that's not the best way to go. So response surface methodology spits out for you the design, produces for you different combinations of the level of the factors and the levels within the factors. So instead of running 81 different trials, you might end up running 24 trials. So different combinations of the different factors and levels, and you get responses. So you get the responses and you plot them against those different independent variables and levels, and you get a polynomial equation, which we call it a prediction equation, that gives you a prediction of where is the optimum response at what levels of the different factors. So, and your response could be either solubility, where do you have the maximum solubility in this case? In other cases, optimal gelation or optimal emulsification. So that is your response. So you get at this optimal high or maximum response, what is the pH, what is the temperature, what's the time, what's the ratio of the enzyme to substrate, and then you can have your optimal conditions targeted toward that enhancement in solubility. And we've done some of that work. For example, for pea protein, we did RSM to enhance the solubility of pea protein. We took the pea protein commercial sample that has very poor solubility, less than 10%. And we subjected it to RSM design and enhanced solubility 6-folds, from 10% to 60% soluble protein. Here's another example where RSM was very handy. So this is work we have done in the lab. One of my former graduate students, a few years ago, we looked at delactosed whey, which is a byproduct of whey production. And it doesn't have, it's low value product, and it has, it's used mostly for animal feed. It does have a significant amount of protein in it, which is 24% protein. And the protein is a functional protein, similar to the protein in a whey protein isolate or concentrate. So beta-lactoglobulin, alpha-lactalbumin. So good, nutritious, and functional protein, but at this low concentration, it's not a functional product. So what we thought about to enhance the value of this product is to make this low concentration of protein functional. So to magnify and augment the functionality of the protein by targeted hydrolysis. So we used RSM. So in order to eliminate the number of trials that we need to do to get to optimal conditions. And we looked in this case at emulsification for use in beef patties, which beef patties, sometimes they add whey protein isolate to it to enhance the water binding and the oil binding of the meat, of the protein. So looking at whey protein isolate, we looked at the emulsification capacity, activity, and stability. And delactosed whey had lower emulsification capacity and stability and activity. And also whey protein concentrate, another reference, also had lower characteristics compared to WPI, which is the protein isolate. So when we did RSM using two different protein enzymes, alkylase and papain, we were able to augment the emulsification capacity at this lower protein concentration so that it was comparable to the emulsification capacity of whey protein isolate. So we enhanced the emulsification capacity. We also enhanced the activity and also the stability of that product. And looking at an application system, which is a meat patty, we added to the meat patty the alkylase hydrolyzed delactosed whey using the RSM design. We compared it to our reference, which is WPI, to control without any added whey protein, and to our papain hydrolyzed, as well as regular delactosed whey and a concentrate. And we found that the alkylase hydrolysis using the RSM had comparable compression force. So meaning the product was more compact, does not crumble easily, and was comparable to a product that we added whey protein isolate to. So RSM can come in handy for targeted modification using hydrolysis. So, however, enzymatic hydrolysis does not always work in the way we anticipate. So we thought that with hydrolysis, we can enhance solubility theoretically. So here in this case, we looked at soy protein isolate, and we looked at hydrolysis using different enzymes, and we saw that we had a decrease in solubility across the board. Regardless of the enzyme that we used, we saw a decrease in solubility. So can you think of reasons why we saw this observation?
There's an exposure of hydrophobic components.
That could be one reason. Another, so from here, sample was boiled to inactivate the enzyme. So boiling the sample, and if you have peptides that you have exposed the hydrophobic interior, you exposed it to heat, then we enhance protein peptide interactions. So so dependent on the treatment, how the hydrolysis was produced, and the processing of that hydrolysis can also impact the final result. Okay, I have lots of background noise. My daughter just came home. The dog is drinking water. The cats are going crazy, so I'm going to move away from here. So give me a moment. All right. Sorry about that. Just got really loud outside. Okay. All right, so this is also to demonstrate what could happen when you have a hydrolysate as well. So here's a protein before hydrolysis, and then you have the protein after hydrolysis. So you might generate peptides of different sizes and may or may not impact the overall characteristics of the protein. So when you heat, you can still get aggregation because you potentially unfolded the protein, denatured the protein, exposed interior moieties, hydrophobic groups, and also potentially sulfhydryl groups. So upon heating, you can also get peptide protein. Intact protein might still remain in your mixture as well. So you might get aggregation via hydrophobic interaction and via disulfide linkages. So we might not solve a problem by just hydrolysis. So what happens when you get aggregation, obviously, even if you have protein intact or peptides present, this can cause problems in different food applications. So in this case, for example, caking of dry food. So baby formula, you will get caking over time, and hardening of protein bars, even hydrolysate proteins or hydrolyzed protein can also cause polymerization over time, causing hardening, which reduces the quality of the product. And if you use it in a beverage, over shelf life, protein aggregation results in turbidity or even sedimentation. So these problems remain unsolved, even with a hydrolysis. So if you think that you want to modify and enhance functionality, you can still end up having issues in a final product. With these issues, you get reduced shelf life, and with reduced shelf life, you have economic loss. Therefore, there is a need to investigate other ways to limit aggregation. So here's an example of an industry problem that is still an issue till the date. Even with whey protein, which is known to be a very soluble protein, the golden standard for beverages. I think I mentioned it last time. Davisco, now Agripor, still have requests to solve the issue of enhancing the shelf life of whey protein beverages. So what happens is, whey protein is soluble, but when you process the beverage to pasteurize it, what ends up happening is the whey protein denatures. And when the protein denatures, over shelf life, it's going to aggregate. So here's a demonstration of what happens. So here's your beta-lactoglobulin, very hydrophilic on the surface like we talked about before. So very soluble protein. It remains soluble at a wide range of pH, very soluble at pH 3.4. However, when you heat it, it's going to denature. You have your hydrophobic interior exposed, your sulfhydryl group exposed, and over time, you will again end up having hydrophobic interaction and disulfide interchange, and then you end up getting aggregation and precipitation. So aggregation is going to increase turbidity of the beverage, especially in clear beverages. You don't want to see that. And precipitation, again, you don't want to see that. So that reduces the shelf life of the product. So especially when you are formulating a beverage that is supposedly a high protein beverage that you can claim as an excellent source of protein. So you want to have more than 4.2% of protein. So the more protein you incorporate in the beverage, the more likely the protein aggregation is going to happen, and the shorter is the shelf life. So even with whey protein, which is the most soluble protein, you have that issue. And so the whey protein is used in acidic beverages as well as neutral beverages. The neutral beverages are generally opaque, like chocolate base or orange cream drinks, whereas the acidic beverages are relatively clear and usually fruit flavored drinks. So the acidic beverages are more desired than neutral beverages because of the fact that with acidic beverages less than 4.6 pH, then you can have less adverse thermal treatment and potentially have better flavor and color of the product with lower thermal processing conditions. So the acidic beverages are favored. However, you want to reduce the pH to lower than the isoelectric point of the whey protein. Although whey protein is soluble at its isoelectric point, but when you heat it, it loses solubility and becomes turbid around the isoelectric point between 3.5 and 5.5, the isoelectric point, and smack in the middle around 4.5. So we want to avoid this range of pH. So usually beverages in the market are around 3.2 to 3.4 pH for whey protein beverages. So they're lower than your 3.5 mark. Then when they're heated, they remain clear. So they don't form the octamers. If you remember from the dairy protein lecture, is that around the isoelectric point, whey protein forms an octamer. But below that, they form mostly monomers and they remain soluble and the protein remains clear. Okay, so what is the problem here? The problem is, okay, the protein is clear in solution, soluble. Other than losing solubility over time, there is an issue with a buffering capacity. So proteins are buffers. So try to reduce a protein solution pH. You're going to need a lot of acids because the protein is going to resist change in pH. So you keep adding acid and adding acid until you get to that low pH of 3.2 to 3.4. So basically, you're adding a lot of acid. And the more protein is in your solution or in your beverage, the more acid you're going to add. And that gives you increased sourness and astringency. So we have two problems here. One problem is the flavor at high protein concentration. And the other problem is with the stability over shelf life, during shelf life or shorter shelf life, basically. So two issues here that they are still looking for solutions. So looking at hydrolysis, yeah, you might increase solubility by targeted hydrolysis. However, we bring other problems with hydrolysis. One of them is, what's your degree of hydrolysis? Is it going to be high that you will be releasing peptides that are bitter peptides? And the higher the degree of hydrolysis, the more the chances of releasing peptides that become more perceived as bitter peptide. So the other issue is with hydrolysis, you actually have peptides, and these peptides are better buffers because now you're releasing more carboxyl and amine groups, so you enhance the buffering capacity of this protein. Basically, more acid needs to be added, then you have more sourness and more astringency. And as I showed earlier, you can still have aggregation over storage. So again, reduction of shelf life. So it hasn't solved the problem. So in this case, with solubility, it wasn't solved. So we need to look at other methods of modification. And one that has been looked at for some time now is Maillard-induced glycation to enhance functionality. And a lot of work has been done specifically on solubility. So in this situation here, what's Maillard-induced glycation? So basically you initiate the Maillard reaction, which is the reaction between amine group, a reactive amine group, and the carbonyl end, which is the reducing end of a carbohydrate, of a reducing carbohydrate. Then it's a condensation reaction. You get your Schiff base and then the Amadori compound, which is the first stable compound in the Maillard reaction. When the Amadori compound, in this case, if you have a protein and a reducing carbohydrate, is your glycated protein. And you want to stop the reaction after the production of Amadori compound. You do not want it to progress to advanced stages of the Maillard reaction, that hence we say Maillard initial reaction is important, inducing the initial reaction and slowing down the propagation to advanced stages. So you all know and have seen the Maillard reaction before. It's very complex. It involves multiple steps and multiple propagators and intermediate compounds that potentially lead to the formation of advanced glycation end products, AGEs. So these could be colorants, brown colors, give you the different flavors, so off flavors, adverse browning, and even polymerized proteins that have lower digestibility and lower functionality. So we don't want intermediate stages and final stages to take place. So we really want the initial reaction and the production of the Amadori compound, and we don't want the propagation of the Maillard reaction. So because the propagation of the Maillard reaction can lead, like I said, to excessive browning of flavor, poor protein solubility due to polymerization, nutrition loss due to excessive blockage of lysine, and also the polymerization leads to reduced digestibility. And there have been studies showing that some AGEs have mutagenic effects, and also potentially all of these negative effects on solubility can lead to shorter product shelf life. So all of this we don't want. What we really want is the Amadori compound, which is the glycated protein. So we really need to control the reaction. In order to control the reaction, so what we do is we run the reaction under suboptimal conditions for Maillard reaction. So we don't want high temperatures. We don't want high water activity, or the water activity which is between 0.5 and 0.7, which is optimal for the Maillard reaction. So we want to be below that. And of course, we want it not to have high temperature for two reasons. One is you don't want to have the temperature high for optimal Maillard reaction, but also you don't want to denature the protein. And we want to use saccharides or carbohydrates that are do not have high reducing power, because when you have sugar with high reducing power, you cannot really control the reaction. The reaction goes fast and it propagates faster because of the smaller molecular weight, and then you have less steric hindrance, and then you have more interactions, and the reaction can go faster, which you cannot control. And you also want to limit the reaction to something we call partial glycation. So if we look at the protein, for example, here is again beta-lactoglobulin is a model that I always use, is the glycation occurs at an amine group. So basically we are blocking an amine group, and most of most reactive amine group is the lysine amine group of the amine group of the side chain of lysine. So we don't want to block all of lysine, and we have a deficient protein, basically. So we want to make sure that it's only partially glycated. That's another thing that we always monitor, is the degree of lysine blockage or amine group blockage. So what happens what does partial glycation result in, theoretically and experimentally proven, is that you get increase in net negative charge. Why? Because we're blocking an amine group that is positive, potentially carries a positive charge. So we block that, then the total net negative charge increases. So with that, we increase surface hydrophilicity, especially when we have a carbohydrate attached, as well as increasing the net negative charge. We are increasing hydrophilicity on the surface. With increased net charge on the surface, then we also have reduced denaturation rate. The higher the charge of the protein on the surface, the more interaction it has with water, the more resistance it would be to thermal denaturation. With reduced thermal denaturation and unfolding, we have reduced disulfide interchange. And with presence of bulky groups, we have higher steric hindrance as well. So we have less likely for protein-protein interaction. We have more water-protein interaction, hence the increased solivation. So with that, we hypothesize that you have, because of all of these reasons, you have better solubility, better thermal stability, and therefore enhanced shelf life at higher protein concentration. So how do we induce the Maillard reaction or glycation? Is experimentally, it's not yet an industrial approach. We are trying to make it industrially feasible, but experimentally, how you do that is you can do that in incubation chambers, which you can have in an industry, incubation chamber rooms, where you can control the relative humidity and you can control the temperature. So we mix the protein and the carbohydrate reducing source together, and we put it in a humidity chamber. Or an older method is we've done before, we had the luxury of incubation chamber, is you can control the water activity in desiccators by putting certain salts that would give you a water activity that is desired. And in this case, like I said, we want it to be lower than the optimal. So we go lower than 0.5. So we put it at a water activity of 0.49. If we have a humidity chamber, then we can put it at 49% relative humidity. Okay, so this is animation that's been done by one of my students. I don't take credit for it. And then we incubate at 60 degrees for which is a temperature that will induce some of the Maillard reaction, but still does not denature the protein. So below the denaturation temperature of the protein, and we incubate at different time points for different time periods, and we assess the degree of glycation. Well, this should have come later, but what I'm trying to illustrate here is that when you glycate, you will have some of the carbohydrate reacting with the protein, and some of them remain unreacted. So you want to end up with a protein isolate. That means you want to end up with a protein that is higher than 80% protein purity. So you want to make sure that you separate the unreacted carbohydrate. And what we've done in the past is separating that by chromatography. And chromatography is a technique that can be industry feasible and is actually used in industry. So we basically separate the non-reacted protein, carbohydrates, sorry, from the proteins and the glycated proteins or the partially glycated protein. Again, a refresher, we use hydrophobic interaction chromatography, which is basically separates based on hydrophobic interactions. So the carbohydrate will not interact with the column, potentially going out with the salt mobile phase, and the salt mobile phase makes the protein covers the charges in the protein, and the protein interacts with the column via hydrophobic interactions. So we get out all of the carbohydrates that did not interact and potentially retain the protein on the column, and then we switch the mobile phase to water. Then the protein will lose the charge, will lose the salt, and then will be eluting with the water. So here is just showing you a chromatogram of a whey protein isolate that showing that the protein comes out mostly with water. And here on the upper graph, this is showing maltodextrin that has not is not reacted, just free maltodextrin, showing that the maltodextrin comes out mostly in with the salt. And then the mixture of a protein and a carbohydrate before incubation, showing also that the maltodextrin mostly comes out with the salt. And the incubated is actually showing that you see some carbohydrate here coming out with the protein that you don't see anymore when you have free maltodextrin or unincubated maltodextrin. And that's an indication of some of the carbohydrate that actually lack covalently linked to your protein. So now it comes out with your protein. So another measure to determine the extent of production of amidori compounds is measuring the amidori compound formation at 304 nanometer. So we measure absorption at 304 nanometer, and we see over time, we see an increase in the absorption at 304, which is an indication of the production of amidori compound. In this example, we were glycating whey protein hydrolysate, and of course we had a control that we incubated without the carbohydrate, and then the sample with the carbohydrate. So we also look at fluorescence. This is an indication of moving into the intermediate stage of the Maillard reaction. So we look at the formation of fluorescent compound, and so that we know that where can we stop the reaction before it progresses to the intermediate stage. So this is another means of measuring the extent of the reaction. Another important, which is most important to us, is the degree of amino acid or free amine blockage, which is an indirect measure of how much of the lysine we are blocking. So we want to make sure that we do not excessively block lysine groups so that we don't lose the nutritional quality. So that is another measure. So we use that using the OPA method of aldehyde method to determine how much of the original free amines were blocked. So we do the assay before glycation and after glycation so that we can determine percent blockage. So this is an example of the effect of glycation on the protein structure. And in this case, in this example, it was whey protein isolate. And we saw that this is a DSC, differential scanning calorimetry. So we look at the denaturation temperature, and we see that after glycation, the denaturation temperature of beta-lactoglobulin, shifted 15 degrees. So that's an indication of an enhancement in thermal stability. Here is a figure showing surface hydrophobicity. So far you've seen a lot, you presented on a lot of papers, or you listened to a lot of paper presentation, and surface hydrophobicity came up over and over. as an indication of denaturation. So in the protein denatures, you have an increase in surface hydrophobicity. So the blue line is basically your whey protein that is not treated, and then the red one is the glycated protein. So we see that the glycation resulted in the protein structure resisting unfolding, and you don't see an increase in surface hydrophobicity. We see mild increase, which glycation causes partial denaturation, partial unfolding. But then you see the regular whey protein, you see an increase and then a decrease. The decrease is an indication of polymerization. With more heating time, the protein eventually denatures and start polymerizing. So you lose surface hydrophobicity because protein are interacting, and hence the surface hydrophobicity is now reduced because protein are interacting together rather than interacting with the ANS reagent that gives you the indication of the hydrophobicity of the protein. Polymerization. So here with what you see is the whey protein isolate, and then heating it, I believe it was like 75 degrees Celsius, and 5 minutes, 10 minutes, 15 minutes, etc. We see that with the more heating time, we see polymerization occurring, which is expected. Here, with the glycated protein, we don't see polymerization occurring even with increased heating time. This is Raman spectroscopy showing the configuration of the protein, the secondary and tertiary structure of the protein, and specific interactions and functional groups. So non-heated and heated. So this is your regular whey protein, non-heated, and then heated at different pHs, 3.4 around the isoelectric point, and then pH 7. So we see that the regular whey protein isolate, when you heat it at all of the different pH, you see marked changes in the spectra. And we have analyzed the spectra and the shifts and all of that, but I'm not showing the tables here. But if you look at the spectra of the glycated whey protein, non-heated and heated, you don't see really any change in the spectra of the non-heated versus the heated. So basically that confirms structural rigidity upon glycation. With that comes enhanced solubility at whether heated or non-heated. So this is heated, and regardless of the pH, we also see tremendous increase in solubility even around the isoelectric point. So the non-treated protein drops out of solution when you heat around the isoelectric point, where the glycated protein remains really soluble. So now why is this important? Because if we formulate a beverage at 4.5, it's still acidic beverage. You can still follow the pasteurization for acidic foods. So mild thermal treatment, preserving the flavor and the color, and you don't need to add that much acid. So you don't have to drop it to 3.4, 3.2 to maintain solubility and to maintain clarity. And then you need to add a lot of acid. So this is a great, outcome, I would say. Another application here is, again, pea protein. We saw that glycated pea protein going from 10% soluble to 100% soluble. So this is another achievement for plant proteins specifically. So this has promise. Now, is it industrially applied? Not yet. We have to still figure out a way to make it industrially feasible approach. One other modification I would like to talk about very quickly here is the utilization of cold plasma. You already heard a presentation today about cold plasma. It is basically used in industry, but not yet so much in food applications, but it's used for disinfection of surfaces and utilization of ozone for water disinfection. wound healing, cancer treatment. So it can be industrially feasible, but we need to investigate it for food applications. It basically, because it is not high thermal treatment, I mean, it does, it's called non-thermal processing, but you do get an increase in temperature, but not really high. But because of that, it preserves quality, it's safe, low energy and costs, efficient, and it's environmentally friendly. But requires that we look to see if it is really beneficial for food application, and at the same time, no toxicity, no toxic components are produced. So these are elements that needs to be looked at. So what is plasma? It's the fourth state of matter. Basically, it's ionized gas, and different gases produce different ionized species. So you have production of ozone, nitrogen reactive species, oxygen hydroxyl radicals, hydrogen peroxide, and you do have some electrons as well. The different species and the composition of these reactive species in the plasma is dependent on the source of gas. So we have air, oxygen, argon, and also it depends on the geometry of the reactor that is producing the plasma. The different chemical reactions induced by cold plasma are mostly oxidation, because you have oxidative reactive species. So you end up getting oxidation of proteins, result in disulfide linkages. You can also get bond cleavage, peptide cleavage, or deamidation, removing of an amine group or removal of a carboxyl group, which can cause changes in the protein ionization status, and also interactions, intramolecular interactions, unfolding of the protein, and polymerization due to disulfide interchange. So basically, these are the main reactions that can happen by cold plasma. So we have done some work on cold plasma. Here is a little bit of the work that Fan did from our group, looking at different configuration of plasma generators. You heard about APPJ today, so plasma jet. You also briefly heard about dielectric barrier discharge, and also we looked at nanosecond pulse discharge. So different configurations. And just briefly here, the main thing we've noticed is that we form polymers, and these polymers are mostly soluble aggregates that are formed upon plasma treatment. We also saw some bond cleavage occurring in APPJ and nanosecond pulse discharge. So these changes are coupled with denaturation. So we saw denaturation at different, all of the treatment caused protein unfolding, and more so with a higher treatment time. With more treatment time, we see decrease in enthalpy, but in comparison to the control, we see major decrease in enthalpy regardless of the treatment, and with more treatment time, we see more denaturation. But with the unfolding and the formation of soluble aggregates, we saw two improved functionalities: gel strength and emulsification. So for example, if we look at DBD treatment for 30 minutes, had the highest gelation strength. And also it was one of the treatments that had the highest emulsification capacity. So comparing it to the commercial pea protein and the native P protein or the control P protein. In all cases, we saw enhancement in gelation and emulsification. Why is this important? Because P protein usually is not as functional as soy, and having a P protein with enhanced gelation and emulsification is really a positive thing for different applications. Another modification, this is one of the last ones I'm going to talk about, is looking at selective breeding, which we talked about as one part of protein modification through selective breeding or biotechnology. And selective breeding, this is an example of work that Vaidehi is doing in our lab, looking at different varieties of, in this case, camelina, and determining which varieties are different in protein profile, and looking at the structure and function characteristics and giving the data back to the breeders, and then they can cross different varieties and different lines for a line that has improved functionality. In this case, Vaidehi selected 4 different lines that have different profiles, significantly different profile, and then she's trying to characterize those, the impact of these differences on the functionality of the protein. So biotechnology is basically, in this case, instead of selective breeding, genetic engineering, where you change the DNA of the organism, which induces some changes in the protein structure, which leads to changes in protein functionality. So it is done, for example, to increase lysine content, so you can enhance protein nutritional quality. You enhance functional properties by changing the amino acid composition or sequence, and maybe promote certain sequences that has physiological activity, so bioactive peptides, that is, or maybe delete sequences that are potentially allergenic. So the protocol here, I know I deleted a lot of slides, but the protocol basically, you want to know the characteristics of the protein and you want to know what changes you want to make. So by doing so, you can get the DNA and modify the DNA sequence so that you can change the protein sequence and composition, basically. And you can use an organism host where you can get the DNA and the modified DNA and incorporate it in the host, and the host will secrete the protein, and then you can characterize the protein. And if it gives you the right change that you're looking for, then you can incorporate that change in the crop or the animal or what have you. So this is, for example, you can do nano or micro scale looking at structure. So penetrometer or penetrometry, looking at structural changes in structural and functional change in a very, very small amount of protein gel, very small. So you can do that. Example application, modification of wheat protein to make it more elastic or less elastic, more viscous by changing the composition or ratio of gliadin to glutenin, or basically changing the sequence within some gliadins and glutenin proteins. To extend the shelf life of milk and avoid the breakdown of casein with the endogenous plasmin enzyme, you can change the sequence where that you delete plasmin sites for the hydrolysis of casein. To make beta casein less hydrophobic, you can initiate a post-translational modification by glycosylating beta casein, which is not originally glycosylated. This is just recap question, quick, before we send you off. So if you have a glycated protein Would the protein be a better emulsifier, a better gelling agent, have good solubility, have improved thermal stability, have lower solubility, have reduced overall functionality, have reduced digestibility, or have improved digestibility? What do you say? From what we talked about. Really quick.
Improve stability.
We could have improved thermal stability, yes. What else?
Good solubility.
It could potentially improved solubility. What else?
Improve gelling.
We could. So we can optimize glycation for targeted application. So we cannot necessarily use one condition of glycation and say we're going to improve the entire functional platform of that protein. We can target, okay, what are the conditions to improve emulsification, to improve gelation, or to improve solubility? There will be different combination of conditions that will result in particular enhancement in functionality. What do you think happens to digestibility?
Didn't you say it could be better? Like
it could be better, it could be worse, depending on your condition. So the more you glycate, so you could potentially hinder the enzyme from being able to access the protein and cut the protein at different sites. So, but if it's partial glycation under controlled conditions, you get this partial unfolding, and some of the studies we've done in the lab actually resulted in enhanced digestibility. So really, it's a very tricky, and there's a lot of controversial research out there, just because degree of glycation would impact whether you increase digestibility, improve it, or actually reduce it. Same with the reduction in quality in terms of blockage of lysine groups. Okay, so here's a hydrolyzed protein then. Very quickly, what do we get with hydrolyzed protein? Or again, hydrolyzed limited hydrolysis, I would say. Like a degree of hydrolysis somewhere between 4 and 10. What do you think happens?
Improved digestibility.
Improved digestibility, that's for sure, because we are breaking down the large protein. We are unfolding the protein in the process, so we get improved digestibility. What else?
Potentially lower solubility.
Lower or higher, depending on the enzyme. We've seen it both ways, depending on the enzyme that you use and the condition of the hydrolysis, and how do you denature the enzyme at the end. So it can go both ways. And same thing, when you want to hydrolyze a protein, you really have to target a certain functionality. You cannot say hydrolyze and I'm improving all functional properties. Depending on the application, you can choose a specific enzyme, and then you can choose different conditions to target emulsification, gelation, or solubility. And that's it. I'm sorry I took 50 minutes, more than 50 minutes. I said 45, but I took more. But at least we're done. And let me know if you have questions between now and the final exam. I'll try to grade everything and post everything by this weekend. And also, Ali and Brigida, please share with me your the collection of the participation points. So I will update everything. I will look at the your practice problems to give you your extra credits. But if you see yourself doing well, don't go into the final not well prepared, because the final is 200 points. So, but at least you know where you stand before going into the final. All right. Thank you all. I will see you later. Bye.
Hey, Pam.
Yeah.
A quick question with the RSM.